The topic of treatment of cancer, I think, is since I don't deal with this every day. I look at the literature every month or something like that, and I find what I think are amazing things because there are so many people working down so many avenues in treatment of cancer and understanding uh, uh, what it actually is and how it evolves. So I thought that it might be appropriate, at least partly for my own learning process, to put together a talk on the evolution of cancer in the context of both how cancer itself changes within a person, within a patient, and evolution in the way that cancer can be treated. And of course, you'd expect that those two things would conjoin as practitioners try to treat cancer. All right, which one are we? That one? That one. Okay, so, I'm sorry?
It's just the
New pants sticker
new pet sticker.
on the side of your leg.
Oh.
Just so you don't walk around all day with it.
Thank you. So I use the word evolution because it connotes changes that happen over time that are inheritable from basically one generation to another. If you're talking about organisms, or from one cell to its daughter cells, if you're talking about simply cell division, which of course would happen as organs grow from the embryo or as cancer develops in the body. So these are inheritable traits. Obviously, that means that they're genetically based. They're selected by the natural environment, and the kinds of selections that natural environment will determine what increases fitness. So if you think about cancer, for cancer to bloom inside of a person's body, partly that means that the cancer cell has found ways to adapt so that it can compete very well with the normal cells in a person's body. So in cancer, this involves mutations that improve the progeny over generations of cell division. Through the evolution of species, it's meant something over much longer time in which improvements similarly have occurred to allow the organism to adapt to its environment. So a successful cancer has genetic changes in it that improve its fitness inside of the body. In order to treat cancer, people have looked at it in an evolutionary way. That is, typically, when you refer to evolution, you start off at some place, and from that place forward, you look at changes genetically in the organism that might start as a branch, as if on a tree, and then you have branches coming off of that main branch that signify significant changes, which would be phenotypic changes, that is, functional changes that would follow genotypic or genetically based changes. So you go through a sequence of these things, and each one of these branches signifies some event or change in the environment of the cell or of the organism that represent basically a difference in fitness. So for example, with organisms it could be a climactic kind of situation. For cells it could be, the instance of hypoxia or some other cell, organism or host response to a cancer. So if you think about cancer itself, and you don't want to be out here after you've had many cell divisions, meaning you have many millions of cells, you'd like to be able to affect the cancer earliest in that whole track of sequences. So you want to, the most effective therapies will target the things that are closer to the trunk before all these mutations have occurred. If you just work out here, you're left with all of the things that were the predecessors to that. And so the goal of all of this is to try to predict the outcomes of the disease itself and of the treatment to the disease. The treatment to the disease doesn't necessarily wipe out cancer, as we know that's not an easy thing to do. And the treatment of disease can merely select for more successful forms of the disease itself. So in some ways it's I wouldn't say hopeless, but it's a difficult task. So and when you think about it on an individual basis, it's even more difficult because there are obviously hundreds of thousands of people with cancer, and it's not sort of a species issue, it's an individual issue because it's something that arises within that person. However, there are some commonalities from person to person within particular kinds of cancer. So cancer cell populations clearly compete with what's already there for nutrients and whatever, and space. And drug treatment, as shown on the previous slide with the tree, if you kill the cell, if you kill cells that are sensitive to a particular drug and you leave other cancer cells behind, then those cancer cells will continue to grow, they'll divide and mutate and do other horrible things. So one of the things that practitioners, that oncologists will do is to adjust the dosage to maintain a balance between cells that they can kill and cells that are resistant to killing. And partly the reason for that is that these two are competing for natural resources, meaning whatever our bodies can provide for them as they inhabit our bodies. So a lot of the treatments these days, as I've come to understand, involve trying to kill primarily sensitive cells that will remain as competition for these other cells. So you wind up with, in the end, tumors that continue to be treatable. Unfortunately, tumors that have to be continually treated. And of course, resistance is a big issue with drug treatment. It can leave tumors vulnerable to other kinds of treatments if you're lucky. People have done what they call evolutionary modeling, and I'll show an example of that in a little bit, to suggest the best way that you can use multiple therapies to try to get rid of these resistant cells. So in order to do all of this, people have spent a fair amount of time trying to understand and measure the evolution of the cancer genome. So the way that works is that one takes early cells in cancer, and those would be what they call the most recent common ancestor to all of the predecessor cells as that clone starts to develop. And do genetic mapping of that and look at populations that arise from that, and you can see by morphology, by genetic testing, by other biochemical testing, you can see where this tree starts to branch and you find multiple populations. And then these populations can be mapped if you look at the normal tissue cells and look at the cancer cells to find specific mutations that have occurred in those cells, and then how those mutations may have advanced. There might be multiples of them. There might be deletions of certain portions of the genome, or there might be repetitions of the same bit of DNA within a cancer in daughter cells of that. So you can have, at this point, one tumor cell population that spreads into two as these events go on. So the focus at that point becomes trying to look at adjacent variants. So you could say, I have these two populations, maybe they need to be treated differently, maybe they can be treated similarly. One of the things that people have done is to draw phylogenetic trees of cancer, and obviously these are, at least in some ways, sort of more temporary or more ephemeral than the evolutionary trees or phylogenetic trees of organisms, because the organisms will stay around for thousands of years, and we don't yet know about the cancer. It definitely evolves in known patterns, but all of those patterns haven't been outlined yet. But there are some patterns that are pretty well delimited. Trying to remember what AML is. Anyway, there are patterns in which the evolution of the genetic material is essentially linear, For example, you start off with a primary cancer and you wind up with only 6 coding mutations, and then as a result of chemotherapy, the cancer will mutate, and now you have 8 coding mutations, and then there's relapse of the cancer that follows. So you can predict that this will happen within a patient in that particular kind of cancer. And another one, this one is acute leukemia, lymphoblastic leukemia. You have branch mutations that can be predicted once you know what you're working with in the beginning. Over time, you wind up with these different ones with different genetic sequences being expressed. So that's a form of branching evolution. Another form of branching evolution that is even more insidious is with breast cancer. If you start off with a fertilized egg, which is particularly a trisomy of chromosome 1q, and that's area Q of chromosome 1. So that means, obviously, trisomy means there's 3 copies of that. And this has specific branching locations in the development of the gene. So if you were to map the gene out, you could say at this particular point, I'm seeing a divergence into 2 populations. And that will continue through the branching scheme. And this repeats itself from patient to patient, thousands of individuals. So having that kind of information means that you can begin to target treatment based on this information. It's not such an easy thing to do because, for one thing, you have to have this massive database, a history of patients that you can line up in that fashion. So you can look at these the somatic mutations that are representative of human cancers, and these are detected in genome-wide studies that typically will involve hundreds and thousands of patients. So one example that I have is in adult and pediatric cancers, and these are just ones that, obviously there will not be a test. We went out. Hello. So just to give examples, neuroblastoma in children, glioblastoma in both children and adults, head and neck cancer, ovarian cancer, pancreatic cancer. All of these transformed cells that are happening in different parts of the body that have been mapped out. The frequency of all of these guys is lifted. I just left this on the slide because it was like this in the journal. I don't expect anyone's going to necessarily read it, but colorectal cancer has the highest rate, lung cancer follows, and melanomas and so on down the line. So someone was looking at this and decided that it would be useful to map this out graphically in a way that illustrated these branching points, and then provided sort of a density map that would give you an idea of where you get this like blossom of events that happens as the next generation of progeny cells from that one progenitor 1, progenitor 2 at each branch point. So if you start off with a normal cell, and at each one of these little stars, there exists something that they call a driver mutation. That presumably is the beginning of trouble, the beginning of cancer, the advent of cancer for that one particular mutation. Not that these have all been mapped out, but certainly some of them have. So after a few of these driver mutations, you begin to go from the normal genotype and phenotype. You start to see changes in the behavior of the cell, its phenotype, its expression, its growth rate. And at some point, you have additional driver mutations that can start a whole other series of events that go on, and these can be predicted from earlier in this point by the initial diagnosis of disease. So within each one of these things, the place where you would like to be having your intervention would be as close as possible to this driver, this most recent common ancestor that is the source of trouble later on. And these things will just continue to blossom until at some point In many cases, you have metastatic cancer. So these can take a number of shapes because there are environmental issues that can happen along the way. Obviously, the cancer is living in the environment of the body. So here's a case with lung cancer where the environment that started everything was tobacco or something in tobacco. And you have various things in the microenvironment that allow, remember that you have the little drivers that happen at the beginning of each one of these things. Some event that is driven by some combination of the environment and the genotype of the individual will happen. And then you have some other big event. You can have a treatment phase which severely diminishes the cancer, obviously, but does it ever go away? I don't know. Relapse at least. And then you have another initiation phase that can happen, and this will continue depending on various environmental factors as well as just the nasty behavior of cancer itself. So there are particular mutations that can drive genomic instability through the cell cycle. An example of these are chromosomal instability, which happens in all kinds of cancer, instability in microsatellites of the chromosome itself that relate to issues with gene repair, and you probably know that gene repair is something that is a constitutive to cells because replication is not 100% faithful. So gene repair is just a normal behavior in cells. Anything that's normal can become diverted into being abnormal. So if you have mutations in gene repair function, that can result in changes in DNA sequence, which obviously may change the expression of the gene. You can have defects in homologous repair of one of these altered genes, which homologous repair usually corrects errors, but it doesn't always. And then obviously exposure to mutagens. So while all of these nasty things can happen, there are checkpoints within the cell cycle. Here's an example of a cell cycle. You start in metaphase, and you have, typical, you have G1, you have synthesis of G2. These are both growth phases. And then you have metaphase, the cells split apart, led by the chromosomes dividing. So throughout these, you have checkpoint genes throughout this entire cycle. You have checkpoint genes at the interface between G1 and S phase. You have at the checkpoint for DNA damage at S phase, another DNA damage checkpoint after synthesis or growth rather in G2, and a spindle assembly checkpoint before a metaphase. Obviously, the spindles in the daughter cells is what pull the chromosomes apart. So you'd like to make sure that that happens as well as possible. But obviously, none of these things are 100% faithful. So as a result of all of the things that can happen, we come down with combination therapies that everybody knows exist. And the combination therapies themselves, they are as effective as they can be, as they can be sort of engineered to be on the moment with a given patient, because it depends on, obviously, on patient response. So here's an example with in a particular case with patients who are responding to this is colorectal cell cancer. So this is a model system for colorectal cancer. This is an antibody, and this is a small molecule drug. Treatment with and this is numbers of tumor cells, basically, over time. The green thing is what happens if you don't add anything. The tumor cells just continue up until some point where they saturate the plate. If you use the drug itself, it gives some reduction in the rate, but not that much. If you use the antibody itself, it pretty severely reduces, but you still get an increase over time. And if you use a combination of the two, then you've wiped everything down to essentially nothing. So that would be a very happy thing to use as a human treatment if you can do that. But obviously, we have to start off with models in the laboratory to do that. So here's a illustration from the same journal where they wanted to show what happens to the cell morphology in these cases. So once again, we have, let's see, these are cells with a combination. The most interesting ones to look at would be in this quadrant of the thing, where you have the combination of the small molecular weight drug and the antibody. They're wiping out these cells in culture at a variety of dosages. So that's a good thing to find in the laboratory. So people have been mapping these things out in some way to try to understand how cancer evolves and how best it can be treated. So in the preceding, in the case of glioblastoma, obviously many things have been attempted, and a lot of drug therapies have been tested. This slide is intended to show the frequency of mutations based on treatment with a drug and with an antibody at specific genetic areas that have been mapped out, like EGFR is epidermal growth factor receptor, for example. Not that I recognize them all, but it's that sort of thing. So these are specific reading portions of the genome, starting off with the initial part, which shows this is the incidence of changes in the sequence, DNA sequence. The epidermal growth factor receptor has a high level of changes initially, and then recurrence, meaning that the cancer has gone away but then blossomed afterwards into full-blown GBM. You have a whole series of different DNA sequences of different genes that are blasting out and being modified. There are certain genes that are in common to both. So in one of the articles that I'm referring, 39 of 58 primary GBM cases changed the subtype, that is the DNA sequences that were measured at relapse. And 5 of these were hypermutated, and all 5 of them switched subtypes. And of these, the mesenchymal subtype on occurrence had worse survival, but the subtype was the most stable, which is unfortunate because the survival was worse if the subtype was less stable, perhaps. So that would not happen. But it turns out that the dominant clone at the initial diagnosis of glioblastoma is usually not the ancestor of the dominant clone at relapse. So that makes the whole business of genetic mapping kind of problematic. You can't just start off and send it to the genetics lab and have them know what's going to be happening with this patient necessarily. Okay, so unfortunately the conclusions are not horribly conclusive, or not wonderfully conclusive, because there's a ton of work to be done. The kinds of work that's been done, a lot of it has been really intensive, high throughput mapping. The kinds of things that require large facilities with a lot of people and expensive instrumentation, and typically major medical research centers. But what we do know, and probably everybody here already knows it, is that there is significant tumor heterogeneity, both in the primary and relapse tumors, that although knowledge is increasing, we can't specifically state in any individual case, let alone generalize across all cases for a particular diagnosis, what are the drivers of tumor progression, but we know that some of them exist, and we have some ideas of what the probability of each one of them is in presenting, in working towards a relapse of cancer. In terms of understanding what's going on more of a fundamental basis, we don't know what the basic parameters of the genetic drift are. That is, we know that we start off with something whose DNA sequence can be mapped in genes of interest, and we know that we can take samples from a patient as time goes on and see how these tissue samples have changed through the patient's treatment and through the course of disease. But what it is that drives this so-called genetic drift is not formalized. It can't necessarily be applied to the next patient with the same diagnosis. And sort of, in a way, good news, but bad news for all of us, is that there are many opportunities to get to understand the basic science that underlies this and to develop better treatment parameters for patient treatment. And those are my references if anybody wants them, and I can email them all to you if you